So let's start. Let's switch to English now. Hello, everyone. I would like to thank everybody who has joined this seminar today. It's a pleasure to have you guys here with us again. So today we are happy to have with us Professor Henrique Marques-Souza. He's an agronomist from the State University of Londrina and have a Master of Science from the Center of Nuclear Energy in Agriculture from the University of São Paulo, and a PhD in Genetics and Functional Genomics from the University of Cologne, Germany. So from 2008 to 2010, he was a postdoctoral fellow at the University of California, Berkeley, at the Howard Hughes Medical Institute. And he has been an associate professor and head of the Department of Biochemistry and Tissue Biology at the Institute of Biology at the University of Campinas, UNICAMP, since 2010. So in his career, he has been dedicated to understanding the function of genes essential to the development, physiology, and survival of organisms considered agricultural pests or pathogens. But more recently, his group has described how the mTOR autophagy pathways are correlated with the SARS-CoV-2 infection, dictating the disease severity, and it's about it that he's going to talk for us today. So thank you so much, Professor Henrique. Feel free to start a presentation. And if the audience have any questions, you can say them directly to Professor Henrique in the end or put in the chat. So we will read at the end, okay? So feel free to start, Professor Henrique. Thank you.
Thank you, Luisa. Thank you, Professor Gustavo, for the invitation. It's a pleasure for me to talk to you about this research we did during the pandemic. And I don't work with viruses and I don't work with immunology, although I have very good friends here in UNICAMP that do. And my expertise is to work with RNA, so gene regulation and RNA. So, but the time that my presentation is going to be a story that started, it has like a clear beginning. And it has also a clear end because I don't work in the field. So I'm going to tell you a story of our discovery up to the point that we could conclude something, right? So it's a ongoing project, let's say, right? So this all started because when pandemic hit us here in Campinas, we have the Professor José Luis Modena, that is a specialist in viruses here. You probably know him. And he was the one that he had a laboratory that was the level 3 biosafety laboratory, and he was able to cultivate the viruses here. So, and when the experiment started to be performed, one limitation, one thing that we found that was limiting our study was to identify the virus inside of infected cells. So the most used cell was this viral E6 cell line. And if you want to know if the virus is inside of the cell or in which organelle or the amount of virus that is inside of the cell, you need to look at the virus, right? So then at that time, I had it was a student of, we say, scientific initiation, right? So undergrad student in my lab many years ago, Luana Nunes Santos, and then she did her PhD in USP with Xavier Neto, and she was going for a postdoc in the United States. This was beginning of 2020, and then she came to my lab, and she was here in Campinas, and she told me, like, look, I'm there. I won't be able to start my PhD. Can I join your lab? And while she was in my lab, she was working with the technique that's called in situ hybridization many years ago, immunohistochemistry and in situ hybridization. So I told her, like, okay, so let's develop a technique to detect the viruses inside of the infected cells, like through in situ hybridization that was way more technically challenging, and also using antibodies for the viruses to do immunohistochemistry, or what we call immunofluorescence. So then we developed this technique. So Luana and the team here in my lab, we developed this technique to detect the RNA-dependent RNA polymerase gene of the virus. So this was the mRNA of the gene. We did a probe for this gene, and we were able to detect this gene inside of the cells with fluorescence. So because of this work, and also comparing this to immunohistochemistry, we were able to detect very specifically the viruses inside of the infected cells. And together with this, while we were developing this, the antibodies for SARS-CoV-2 also arrived. So this on the right is a cell. Those are not double staining, those are different cells, but just to show the mRNA of the virus on the cell on the left, and on the cell on the right was just a spike protein. It's fluorescent antibody stain for the spike protein. We were not able to do double staining of antibody staining and in situ hybridization, right? So, but we saw that it was a very, very similar localization pattern. So because of this, this capacity, then we participated in many works that were published here in the Biology Institute. So with many authors, collaboration. So this was with Marco Aurelio Vinolo, that we were able to detect the viruses inside of the gut epithelial cells using the antibody for spike. And then we saw that there was no difference. Although the literature was suggesting that this would have a impact, the short chain fatty acid would have an impact in SARS-CoV-2 infection, and we showed in this Gut Microbes journal that paper that was not the case. Then we also participated in this work with José Luis Modena, showing that the P1 lineage was not detected using the original antibody. So we used the antibody, and we saw that although it was detecting the initial variant, the second variant, we could detect with the antibody against the N protein, but not the spike protein, the S protein. And then we also participate in this work with led by Marcelo Mori, that's showing that the SARS-CoV-2 would infect adipose tissue as a reservatory of viruses inside of patients, right? But while we were participating in this work, and all of this was done through confocal imaging, this is one of my expertise as well. So I was on a Friday night looking at the cells, and then I started to realize that there was some peculiar staining or staining pattern in the cells. And as you can see here, this roundish pattern of localization, so looking like the virus would be occupying an organelle. And then we talked to many people with Hernandez here in the Institute of Biology, many other collaborators. And we did a panel of antibodies to see which organelle was that. And we found out that the spike protein or the viruses was colocalized with the labeling for P52. So in the affected cells, you see, if you're familiar with immunofluorescence, then we have red and green. So green is the protein of the virus is spike protein, and red is the P52 protein. And the colocalization is in yellow. And P52 is P62, not the band, right? But P62 protein is a protein that occupies autophagosomes. It's a marker, it's a protein that is a marker for autophagosomes. As you might know, autophagosome is an organelle that participates in autophagy, right? And is controlled by mTOR signaling. And mTOR signaling is controlled by nutrition. So those in this scheme here, just showing that on the left, you have like a nutrient rich conditions. You have mTOR signaling bound to the lysosomes, and the lysosomes they become deacidified, so they're not acid anymore. The lysosomes are like a bag of acid, and everything that fuses with lysosomes get degraded, get fragmented, proteins, RNA, and whatever. So then when mTOR is active, lysosome, they're not functional in this lysate, in this for its lysis or function on lysis, right? And in starvation, then you have autophagy, is that when you don't have nutrition coming from your bloodstream, then the cell is able to recycle old and non-functional proteins and through autophagosome. So the autophagosome gets those proteins, the autophagosome fuses with the lysosome. Because mTOR signaling is not activated, then the lysosome gets acidified. And then when you have the fusion of autophagosome with lysosome, then all the protein is degraded. And that this is sent through the bloodstream to provide more amino acids and nutrients for other cells. This has happened when you have starvation and also in other situations that you have a depletion of nutrients in the bloodstream. Very generally speaking, right? Autophagy is a very complex process. I'm just showing the basic to understand our hypothesis. So then it seems that, as we saw, that SARS-CoV-2 might use autophagosomes to replicate in viral cells. So it might use, we didn't show that it does, but it might use autophagosome to replicate, right? So at that time, José Luis Modena told me that another guy that I have never heard about, that Fernando Cimabuco, he was a colleague here in UNICAMP at that time. Now he's a professor in the Department of Biochemistry in Unifesp. He was also looking to mTOR signaling in the virus infection. And Fernando is an expert in mTOR signaling in cancer biology. So then I explained him what we were seeing, and then he said, okay, so if we're doing this, we have to look to many other proteins to see how mTOR signaling is being modulated in infected in virus in the SARS-CoV-2 infection, and also proteins related to autophagosome, like those two LC32 and P62, 62. So then he lab did all this. His lab did all this experiments, and then he saw that many proteins that were marker for mTOR signaling, like the phosphorylated version of S6K1, was increased, was accumulated in viral infected cells compared to the mock, to the control. Also, PS6 phosphorylated version of S6 protein was also accumulated. But then when he looked for the proteins that were marker of autophagosomes, then he saw that with the infection of the virus, you had an accumulation of the autophagosome proteins. And this is the two versions, that LC31 and 2. And when it's included in the autophagosome, then it is the second version, the LC32. And also with P52, it's showing that the autophagosome, the vesicle, the autophagosome compartment, organelle, was accumulated in the cells. So if you have those protein and autophagy flux is working, then those proteins, although they are markers for autophagosomes, they are a negative markers for autophagy flux, because they are degraded when they fuse with the lysosome. So if autophagy flux was happening, then you would not see the accumulation of those two proteins here. So it seems that something is going on with this step of fusion, right? So it could be that the lysosome is not acid, because we see activation or we see accumulation of mTOR signaling. So it could be that they are not degraded because they are not being degraded by the lysosome, or it could be that even though we expected lysosome would not be active because of mTOR, it could be that there was a problem with fusion as well. So then what we did was to do a simple double staining with the virus, with the lysosome marker LUMP1, and you could not find one yellow pixel in this image, right? It's either red or green. So we could not observe any colocalization of spike protein labeling and lysosome marker LUMP1 labeling. So it could be that, or it seems that viral or SARS-CoV-2 infected viral cells accumulate mTOR and autophagosome markers. That's a fact. And also SARS-CoV-2 containing autophagosomes do not fuse with lysosome in viral cells. But this was all viral cells, and we wanted to know what was going, if what we were observing at that time was also happening in humans. So at that time, then we started a collaboration with Elder Nakaya and his postdoc at the time, Thomas, and then they were analyzing, they found and they were analyzing a data set of single cell transcriptomics in COVID-19 patients. And because it was single cell transcriptomics, we were able to separate this patients with COVID-19. We were able to separate cells from patients, or cells that were infected in patients with COVID-19 and cells that were not infected. When you get the viruses, it's not all your cells that are infected, right? So this data set had this difference. So we had the control that was patients that would not have the viruses, would not have the infection of the viruses, and what we call bystanders, that are the cells that are from patients with COVID-19, but the cell did not show RNA from the virus in the transcriptomics. So, and what we saw here is that when we compared bystanders and infected cells with patients that did not have COVID-19, we saw, just like we were seeing in viral cells, an increase of mTOR genes, related genes, a decrease in autophagy related genes, and an increase in negative regulation of autophagy. And that could mean two things. If you're comparing infected cells with the control and bystanders with the control, it means that all the cells from a person that had COVID would have high mTOR and low autophagy, or a capacity to do autophagy, right? So it could be that the viruses infect those people and deregulates mTOR and autophagy in all other cells, like a systemic response. But also it could be that those people with COVID-19 severe symptoms, they already have a high mTOR signaling and a low capacity to do autophagy. So we didn't test for that, but those are the two hypotheses that we were working. And if you compare infected cells and bystander cells, you don't see difference. So it still could be both things, right? But when we compare infected cells with bystanders and look into lysosome genes, we see a reduction of gene related to lysosomes. So it could be that we could say now that something specific for the virus's infection. So the cells that have the viruses inside of the cells, those cells repress, or just in those cells, lysosome-related genes are repressed. So, and if we look for the gene that codes P62, that is the SKSTN1, as the gene that is called P62, we saw that in patients, just as we saw in viral cells, in human patients with COVID-19, you have an accumulation of autophagosome marker genes. So it's also indicating that you have the situation that you have high mTOR and not depleted, but reduced capacity to do autophagy. So therefore, you have accumulation of autophagy. So SARS-CoV-2 infection in humans might benefit from a state of high mTOR signaling and impaired autophagy. So, and at that time, then I started seeing what was the relation between mTOR signaling and the comorbidities that were found in COVID-19. And then I found this paper that was showing the 10 more frequently found comorbidities in people that died from COVID-19. Those were the 10 top ones, and there were only those 10, right? All of them, without exception. And then I looked in each one of them for papers, and in all of them, they were described already that they have a chronic activation of mTOR signaling. So then it could be that in all those patients that they have severe and they ended up dying from COVID-19, they have a situation that mTOR signaling is high, and therefore you can predict that lysosome would be not functional, autophagosome would be not degraded, and then if the viruses is using autophagosome, then it would benefit, right? The viruses would be a benefit in this patients or in this health condition. So, and also we know that at least in the beginning or the highest peak of pandemic, we have 80% of patients hospitalized, they were elderly, and increased mTOR signaling and hampered autophagy are hallmarks of aging. So this also in line with our findings and our hypothesis. So, and then we were looking at this process of lysosome and lysosome fusion to see, we saw that lysosome related genes are downregulated in patients, right? But was there anything related to fusion? So when we looked at this data set, then Elder Nakayan and his group, they found, and Thomas, they found a gene that is this VUMP8 gene that was the most affected gene in the data set that we are analyzing with this VUMP8. It was very reduced in the infected patients. And VUMP8 is not only a gene, it's called a snare, that is a gene that is important for fusion, for vesicle to vesicle fusions. Then here showing VUMP8 is important for the fusion of autophagosome to lysosome. So it seems that the virus is reducing a gene, the expression of a gene that is important for the fusion of lysosome with autophagosome. So it seems that it is a backup. So it's in people that has high mTOR, lysosome is already not so much functional, right? So it's not a 1 or 0 situation, but lysosome is depleted, let's say, the function. But the viruses also affect the fusion. So even in the situation that lysosome might be functional, then the virus would try to guarantee its success on using autophagosome by reducing this gene that is important for fusion of autophagosome and lysosome. But I also found that this gene is not only important for vesicle, but also vesicle membrane fusion, and showing that VUMP8 is important for at a certain level secretion of cellular content to extracellular environment. So then one idea is that when I saw that, I said, oh, well, could it be that by reducing the expression of a gene that would prevent vesicle fusion, that is important for the viruses, because by preventing this, the viruses on autophagosome would not fuse with the lysosome. By reducing this gene, the virus might also be avoiding secretion to the supernatant. So it could be that the viruses try to do something that is like preventing fusion with the lysosome, but it would also end up preventing its own evasion from the cell. So, and one meeting that we were having here with Luis Modena, Marcelo Mori, Daniel Martins, Pedro Vieira, Marco Vinolo, Alessandro, we were having this conversation, and then Modena said, like, oh, do you know, we were having a lot of trouble having a high load of viruses when we were producing the virus, because they were producing the viruses for the experiments, right? So then it said, like, but then we were collecting the supernatant. So, but then one day I just decided to do a cell lysate, and then we had lots of viruses. When he told me this, I chat with him privately and said, like, okay, do this experiment. Count the cells, take only the supernatant and count the viruses in the supernatant, and then take only the cell lysate and count it. And the basis is that SARS-CoV-1, the virus that caused the SARS epidemic in 2003, it is found 10 times in higher numbers, 10 times more in the supernatant than inside of the cells, right? This is SARS-CoV-2 precedent, a cousin, right? And when he did this counting, we saw that in SARS-CoV-2 were found 20 times more viral particles inside of the cells. So instead of having 10 times outside of the cell, the supernated, in SARS-CoV-2, at least in the conditions that we were raising the viruses here, you would find 20 times viruses inside of the cell, as if the virus were being held inside of the cells, right? So then it could be that SARS-CoV-2 infection reduced the expression of proteins related to, or the expression of genes and therefore proteins, right, related to vesicle and vesicle membrane fusion that might, we saw already, prevent lysosome to autophagosome fusions, but also prevent SARS-CoV-2 containing vesicles to fuse to lysosome and to evade infected cells. And what is it's interesting that I look back to the stainings, right, to the microscope, and then I saw that there's lots and lots of cell connections. Those are epithelial cells, so they are connected, right? They're normally connected. The cells not infected, they are also connected. But we saw lots of viral particles like this image here. This was the first image I saw showing a possible cell-to-cell transmission, right? Those are the particles, the viral particles that I showed in the beginning, this round particles here. So, and you can see a cell that is lots of virus here transmitting, or at least having, I cannot say that it's transmitting, but having viral particles in the cell-to-cell connection, and you see those particles in this other cell. So we didn't do any live imaging to see the direction of this infection, but you could see here clearly that there is genetic material of the viruses in the cell-to-cell connection. So, and it's interesting because this is a graph showing the amount of the genes, the viral load counting from the day of the symptoms onset. And differently from all other related related viruses, viruses related to SARS-CoV-2, SARS-CoV-2 would have a huge viral load before the day that you have the onset of the symptoms. So then it could well be that the virus is accumulated inside of the cells, and the symptom is a sign that your immune response saw the danger is now leading with it, right? So it could be that indeed the case that the SARS-CoV-2 is locked inside of the cell and also not being degraded by lysosomes, so it's accumulating, and then when it releases the cell, maybe when the cell dies, then you have a lot of viruses coming out, and therefore you have a very strong immune response, because differently from other viruses that use the cell to replicate, and as soon as they replicate, they leave the cell, the immune system starts seeing the viruses coming little by little, right? It could be that in this case, what happens is that the viruses accumulate, and then when the immune system sees the viruses, it sees so much that the response is brutal, right? Like we see. So it's something that's not, we didn't study this, but we're just correlating with what we saw in the literature. So then, and then we did double staining for this organelle to try to find what was this round thing. This was not the P52 labeling. This is much bigger than the P52 label, and we saw that it's positive for CD63. So all those vesicles here going from one cell to another cell, they are all CD63 positive labeling as well. And interestingly is that CD63 is a marker for MVB, so this multiple vesicle bodies, and also late endosomes. And when you have the fusion, I found this paper showing here that you have autophagosomes, autophagosomes and those MVBs and late endosomes, they fuse, forming this amphysome. And then amphysome is positive for CD63. And then here is showing that using a snare, a protein that is important for vesicle membrane fusion, then the content from this amphicone will be released to the outer environment of the cell, or would be secreted then. So also this is very speculative. We didn't test for that, but it could be that this is all happening in the cell, but then the viruses prevent also all those vesicles to fuse to lysosome, but also that is here, right? So it prevents this, but also when it arrives at this point, it prevents the amphicone fusion to the membrane to release the virus. So then the amphicone containing the virus containing amphisomes would be stuck inside of the cells and then would pass through the cell-to-cell connection to the neighboring cells. So this is a scheme that we made, and when you have low mTOR signaling, then you have autophagosomes, and you have this MVBs, then you have this amphisomes, or both of them fusing with the acidified lysosomes, and then you have a release of the content. And the content of this lysis, right, would be a result of this lysis would be the content of the organelles that might have the viruses. So you get infected with the viruses enters the cell. We haven't explored this mechanism of entering the cell and also start replicating. And then when it's inside of this organelle and you have lower mTOR, and if you're starving like you were today, because now it's lunchtime, then it could be that the lysosome fuses with the either autophagosome, late endosome, or the fusion that is amphicone. And then if there is a virus inside, the protein gets degraded because the lysosome is acidified, and then the pieces of virus protein get sent to the out of the cell. And then if that's the case, then you might develop antibody if the proteins of the viruses get outside of the cells, right? But when you have high mTOR, either because you're eating very frequently or because you have one of those comorbidities that increase ectopically the mTOR signaling, then what could happen is that high mTOR would deacidify lysosomes and also prevent vesicle fusion and vesicle membrane fusion. So then the virus would not be degraded by lysosome and would now also not be secreted from the cell and start accumulating, pathing to other neighboring cells, and then it will evade the cell when the cells die. We haven't tested for that, but that's one of the explanations. And that's it. That's the hypothesis that all started with Professor Modena here, and the project itself started with this imaging that we saw this cell-to-cell connection with viral particle, and then we started looking at it and put together a very nice team of researchers and came up with this story, right? It's very correlative, right? Because we didn't have time or expertise to go deeper, but that's a hypothesis that matches with what we observe for the effect of this virus in humans. So we hope that other people would explore further this work and see if that is the case. And if that is the case, then could explain why, just finalizing, it could explain why differently from any other gene we saw in history of humankind, different from any viruses, that were more successful in countries that are well known from their plentiness of the food, right? Brazil, United States, Italy, Spain, France. When you think about those countries, you think about food all the time, eating all the time, right? And then you wonder why in countries of Africa, or even like people living in the street there that are often, they suffer a lot by viruses infections, many viruses infection, why they were not more affected by this infection, right? So this is all a scenario that, based on our work, we believe that a situation when you have low mTOR and autophagy working properly in your system, then if you get the viruses and maybe you stop eating, let's say at 5 p.m. and then you eat again at 9 p.m., you do like intermediate fasting, it could be that autophagy flux would eliminate the viruses that get infection, and then maybe you even can produce antibody without having any symptoms. But if you're not used to do autophagy for any reason, then it could be that the virus would use this organelle, and also the fact that the organelle is not being degraded and be more successful in replication. That's it. Thank you very much.
Thank you, Professor Henrique. Amazing talk. It was very interesting. So now we are open to questions. You can put it on the chat or raise your hands. So if anyone wants to start to asking for Professor Henrique. Gustavo?
Yeah, just to warm up things for the students. Henrique, great. This is really cool story. Has a lot of implications, as you mentioned. Some need still, further work. I have just one specific question related to things that I've been working on, which is the CD8 T cells. So, do you know? We know that the CD8 T cell function is impaired in some of the patients. Now you mentioned that the virus may interfere with the lysosomal formation and the transport of vesicles. The cytotoxic granules are lysosomal. So does anybody look at this aspect in the CD8 cells? First of all, are they normally affected?
Yeah, that's a great question. We did, this was for one of the, maybe the first study that we did before even discovering this, was to do in situ hybridization and antibody staining in CD8 cells with Alessandro here. And we found the viruses, we found the RNA and also the protein inside of the cells. And oh yeah, we did double staining with CD4, and we did, I think we did CD4 and other cells, but then we saw the cells inside of T cells. I'm not totally sure all the markers that we did to see in which cell, but also this single cell. I was thinking that single cell transcriptomics, which type of cell were there, but they were epithelial cells, lung epithelial cells. But I'm not sure if we could take a look if there were other cell types, specific cell types, to see if the genes related to lysosome would be reduced there, because you could see both things. You could see in the single cell transcriptomics if the CD8 cell would be infected. That's what we saw, and also if they would be reduced for the lysosome. The in situ and antibody staining would not help, because what we saw with the expression, and I'm not sure, I don't remember the lump one. I don't remember seeing any difference in amount of protein, amount of staining that we.
Yeah, but not. You wouldn't see, I think, a LAMP1 expression, but you could see externalization. I think you have all the setups to just incubate the virus with the CD8s and then stimulate the CD8s and see whether, the granules are more, or, there's some impairment of formation, and then the degranulation, because I think this would be also a cool thing. Perhaps some of the people that has difficulty to resolve the infection has impairment in the degranulation of CD8.
Yeah, interesting. Just by looking at that would be the mechanism why the cell it's not functioning, right? And then if that is the case, then you have already a hypothesis for the mechanism because the gene reduces the... Nice.
Thank you.
Bruno?
Hi, Luisa. Hi, Henrique. Very nice talk. We are still having the students warming up with some questions here, but we do have a question from Professor Leonardo Travassos. So, come near the microphone.
Hi, Henrique. Really nice talk. I was wondering that maybe the accumulation of P62 will not be the best way to look at an interference of the virus with the autophagic flux. Maybe experiments with cells infected and cells infected plus the presence of, I don't know, chloroquine or something like this. And I'm telling that because we have evidence that, I mean, different setting, but we can have an increase in autophagy and an increase in P62. So maybe P62 will not be the best marker for autophagic flux. My second question is, do you have evidence of the presence of the virus inside autophagosomes? Or, I mean, a localization of LC3 and the virus?
No, the antibody we have for LT3 didn't work. We tried a lot of times.
It's very,
So when you say that, related to the first related to the first question, when you say that there is an increase in P52, you say protein content.
Yes, yeah.
Okay,
protein,
so the autophagosomes
an increase in P62, even if, for example, we have evidence of inhibition of mTOR, so we give rapamycin the cell, and we have an increase in P62. So situations in which we have an increase in oxidative stress, we have an increase in P62. At the same time, we have induction of autophagy. We have an increase in LC3. So it's a little tricky sometimes.
Yeah.
So
yes,
I would suggest you to have a condition in which you infect your cells in the presence of chloroquine.
Yeah.
That would be the best way to analyze autophagic flux.
Yeah.
Right.
What do you think about the fact that we also tested for LC3? Do you remember the ratio, the LC3 1 and 2? I think the expert, the Fernando is the expert in it, but then he was his argument is that P52 might be not totally related, but if you have a combination of LC3, then you cannot have.
Yeah, let's think this way. You have an increase in LC3-2, right? A conversion of LC3-1 to LC3-2. That's a picture, right? But it might be an induction of autophagy, but it could be a blockade in autophagy as well, right? So to clear this problem, I think we need to have baflomycin or chloroquine, so that if you have a further increase in LC3-2 in the presence of these drugs, then we can clearly say that we have inhibition of the autophagic flux.
Nice, yeah. So I actually did that with the baflomycin, but I didn't test for the colocalization. What we saw was that you have more viruses outside of the cell.
Okay. Okay.
Yeah, you have more viruses. So it was not shown that it was fusing But then when we did a PCR, and it could also be that it was fusing with lysosome and sent it outside, and that the PCR just got fragments, but we saw more viruses outside of the cells. And we did also with the, when you use to count the viruses, right? So it's like living viral particles. And we saw that there were more viruses outside of the cell when we treated with bafilomycin. We wanted to do some chloroquine, but then it was very political, so we didn't get into that.
No,
But I believe you're right. I mean, the best way to do.
I think bafilomycin is okay. Bafilomycin, chloroquine sometimes it's very harsh for the cells. Bafilomycin, I think it's more gentle. I mean, it's an easy experiment to be performed.
Yeah, nice. What we could have done, I was thinking now that we could have done, based on what you're saying, is to do that double staining with the virus and the lysosome, right?
Yeah, well,
After
that's
treatment. Yeah, nice.
Okay, well, thanks a lot.
Thank you.
We do have another question here from a student. If you go ahead. Is it okay, Luisa? Yeah, okay. So Lucas, please.
Good afternoon, Henrique, and thank you for your talk. It was very interesting. I must say I'm not an autophagy expert, so I was wondering actually if you have seen an increase, since you have increased virus accumulation inside the cell. Have you seen an increase in other forms of cell death, like increase in necrosis, apoptosis, other forms of cell death? Or if the viral spread, like the cell-to-cell spread, is enough to prevent the cell from dying from the accumulation?
Cool, nice question. We don't believe that the virus would be preventing the cell to die. We don't have any evidence for that. It could be that there is a mechanism that would prevent apoptosis and so on, but we didn't touch that. When we saw, I wanted to do an experiment. We thought about many experiments to see if the virus would induce the connections, but we didn't. We tried many things, live imaging and so on. But the thing that I can say is when you compare non-infected cells and infected cells, you see more or less the same thing comparing connections, right? So it seems that the virus is not modulating that, but we're not conclusive on that. But when you start looking through the cells, you see many weird things. I have many images that I cannot explain, but they are there. You have, and things that is common to virus, we have a polynucleated cells, like a giant cells with like 18 nuclei. And you don't see this in uninfected cells, right? But then I said, well, if you want to show this, you have to prove and so on. So we didn't look to that. And there were people also studying with the syncytial, and this is something that viruses do, right? So, and also many cells that are dying way more than the non-infected cells. This is normal. This is what you expect. But we didn't see any specific thing. It's just things that cells are dying. And when the cells are dying, it's difficult to see because when they die, they get autofluorescent. So when you see a very dense cell, it's autofluorescent. Sometimes it's autofluorescence, or most of the times. So, but you could see the pattern of the virus, of the presence of the viruses. So you could see that the cell is like the DNA starting to fragment because of the DAPI imaging, and then you see the cell is loaded with viruses and it's dying. But nothing is specific, right? So, okay, we see this amount. We didn't test for that. We just saw that the cells are dying, and they were some weird cells forming, like this multinucleated cells.
Okay, thank you.
Thank you for your question.
Bruno, can I ask?
Sure. We actually have another question here from Rio.
Sure.
So, Professor Schaffenstein, please.
So
Congratulations on Fluminense first.
commemorating the victory. So I unfortunately I came too late to your talk. It's too sad, but tomorrow I'll see the day. Question regarding antigen processing, class 1 restriction. How does this dichotomy affects actually the processing or also the expression levels at the surface of class 1? Because since you have a diversion, I mean, you just tend to think about the CD8 T cells, you know, coming in at a certain point. And it is very important for the virus, you know, to evade the system at these points. But what are the facts? If you compare one situation versus the other, how the CD8 T cells are seeing, you know, this strategy of evasion that you have described?
Yeah, I'm sorry, I have no idea whatsoever. I mean, I know the work, I'm not immunologist, and I know the work from the guys here, right? So the role of those cells, and as Luisa told me, I'm an agronomist, so I not even studied that. But I might be simplistic, and I might be simplistic that to think that the cell would be hiding the viruses, or the virus would be hidden inside of the cell. So what happened after that, and what happened the process? So it could be, and you correct me if I'm wrong. So it could be that the cells just like they die because they have so much or so many viral particles inside that they die. And then when they die, the virus is just out. Or it could be that there is a mechanism of secretion, but we didn't touch that. All the things that I said about fusion is speculative, is based on the reduction of a gene that is important for that. And the fact that we didn't publish this in Nature Cell Science is because we didn't follow up those questions. They were very important questions. So we didn't have legs to do that. And so those are very important questions, but we didn't explore this. And my idea as one of the authors or leading author of this work is that I want the story to be out so people can go there and ask questions, right? So they can challenge this and then can show that this is like all nonsense, you know, or not. They can say, you know, if we would have known this the first time that you submitted the paper, then we are fighting for like 1.5 years to try to publish this work, right? So maybe if we know this evidence, we could have asked so many other questions related to this relation between autophagy and so on. But I think that the immune system I see as the immune system is out of the work. It's just there waiting, right? So it doesn't matter if it's secreting or if the cell is dying, and after that, then the immune system would take place. That's why I didn't have we didn't explore immune response in the system just because of that. This is an assumption, and we might be wrong.
Yeah, Gustavo is in an excellent position to help you answering this.
I'm not in an excellent position, but I can try to contribute. Although I must say that you know, I may say things that is wrong.
Anything you say is going to be more precise.
No. I feel that the transport of proteins from the Golgi to the cell surface is intact. I don't remember seeing anything that supports that the fact that proteins will not be expressed in the cell surface, including the MHC class 2. I think you are saying something very particular with the lysosome, autophagosome, lysosome pathway. And the virus, as far as I understand as well, do not replicate in the phagosome. It needs the machinery in the cytosol. Okay,
Yeah.
so I think the antigen presentation should be relatively intact,
I think so.
but then the virus accumulate and maybe transfer by this very elegant system that you described. That's what I think, but I may be wrong.
Yeah, I would say so.
But by the way, Julio, very nice shirt. I'm very happy for the tricolor from Rio de Janeiro, because my tricolor here is not doing so well.
Well, your time will come. Okay.
So Yan asked me if we try to inhibit mTOR signaling. So we did with rapamycin, and we had unconclusive. So depending on the viral load, so there was something complex that we could not address that. We wanted to try to inhibit mTOR signaling through something more specific, but that would be something after to try as well. But just treating with rapamycin, we one experiment, we saw exactly what we wanted, we would expect. But then the other one was opposite. So, and then the guys were also trying to inhibit here to see if you would reduce infection, and it was the same thing. The lab from Marcelo Mori, they had contrasting results, so we didn't go further with that.
Henrique, I was about to ask some very similar question to Jan's question, because when you talked about the more activation mTOR, the COVID-19 patients, I was thinking about the in vitro experiments. If you modulate the nutritional status of cells, thinking about the activation of mTOR, you see more lipids or more glucoses. Do you think that this autophagy pathway could work differently? And also the viral load?
Yeah, so we tried to do this in another cell, but the problem, because VeroCell is depleted, or Gustavo might know this better, depleted or mutated for interferon. So then all this response, interferon-related response, is not normal, let's say. And we know that interferon is also affecting mTOR signaling. So we didn't want to. I proposed this to Fernando. Said like, well, let's just like deplete, right? Let's starve VeroCell. Then he said, well, it wouldn't work because the cell might be like the chronic activation of mTOR signaling in those patients, right? So maybe even if you know this and you want to do intermittent fasting, for example, if you have chronic activation of mTOR, it might not work. So then when we tried, then I said, okay, but let's try to modulate this in vitro, like you were suggesting. So then we wanted to go to another cell, but then the cells would never infect like VeroCells. Even Tiago Cunha, he had which cell line was that? That was overexpressed in ACE2 to see if it would infect more. It was a human epithelial cells. I forgot. I think it did it for many cell types, but he provided me one of those cells, and then we see if we would have more infection because it was increased for ACE2, but it didn't infect. So that it also somehow correlates the fact that VeroCell might be a more suitable cellular condition or situation for this infection. So it's not only by having ACE2 that you would be infected, right? You overexpress ACE2, now you have lots of infection. No, you might have lots of viral entry in the cell, but that doesn't mean that the virus would replicate and would affect you like very. So it needs mTOR signaling. Every virus, we need mTOR signaling, and everybody does. Every virus also do, because mTOR signaling is for protein synthesis, right? So it needs this increased mTOR signaling, but also this depleted autophagy flux zone. So we didn't have the in vitro system to test that, although we looked for. And that's why it was so important to see if that was the case also in humans, and it was the case. So it correlates. We haven't done any. I wanted to do a clinical trial with, like things that induce autophagy, and see if people would be, not less infected, but less with. It's just like the vaccine, right? So, but the vaccine is after that. The vaccine would just like get the viruses coming out of the cells. And maybe with people doing like intermittent fasting, they would, they would eliminate the viruses. They would be infected, but not having symptoms because the virus would not accumulate in the cells.
Nice. That's very interesting, Henrique.
Nice.
So,
I hope it has more work coming out of this.
yes. Do we have more questions on your side? So I think we'll have no more questions for now, but it was an amazing discussion.
Anderson has a question. Lisa.
Oh, he's clapping hands. So thank you so much, Professor Henrique, for being here with us today. It was a very good discussion with us. So I'd like to thank you in the name of the organizing committee. Thank you so much.
Nice, thank you. Thanks a lot. It's the first specialized crowd that I talk about this work. It is very nice. It's a story that I'm in love with. Although I don't work with that and I'm not going to work this any longer, it's something that if I could clone myself, for sure, this is a work that would go down through 10, 30 years studying this. This is really nice. It could be also that it's something that could be somehow at certain point common to other viruses, right? And if you think before we have refrigerators and fast food and iFood, we would not eat after 5 p.m., right? We didn't have fridge. We had to find shelter. So we would starve. We do intermediate fasting every day, just because we were like one animal in nature, right? So it could be that in our history, we were protected by viruses in some aspect, just because every night we were just clean our system by recycling proteins and whatever is used in our autophagosome. So it's nice. It's a fascinating story that is still very correlative, speculative, but it's promising.
It's very good, very good. Thank you so much, Henrique.
Thank you again for the invitation, and